Imagine a situation where you were to influence a mass amount of people, be it by, well, actually, look more at a more realistic situation, a tad more abject. Despite your knowledge or backing on the topic, you may not be able to have that conclusion come about, because a lot of people are not familiar with recombinant gene technology and how it underlies different like GMOs in the common debate. So what I would like to do is, I suspect as college matriculates, your persistence in pursuing education is not based on elating, say, like pecuniary disposition, but more as like a consistent pursuit of bettering your knowledge. So, what I will attempt to do is provide a foundation for you to better approach and decide whether or not it's appropriate to consider different positions when they arise in different contexts. I will do this through three different ways. First, I will start with horizontal gene transfer in plasmids. I will then go to that application for a while, and following that, I will kind of debunk some of the misconceptions surrounding it using the previous two methods. But prior to that, I should establish credibility. I am currently doing my biomed undergrad here at MSU. Though I've only been here for a year, I have taken a few upper division courses in my major, including recently, this previous spring, I did a Bio 499, as well as just recently Bio 470, so I just kind of bypassed generals. As well as that, I'm also working in Dr. Sorensen's lab, where we research, it's pretty much a parasite-host coevolution, so I'm slightly familiar with science. But alright, going to the first topic. With horizontal gene transfer, that can basically be defined as genetic transfer within a generation, as opposed to vertical, which is what you see like in sapiens, where copulation, combination of the genetics in an offspring. This would be more so, actually we'll focus prioritize bacteria. We'll start with transformation. Transformation is a type of horizontal gene transfer which was shown to be true by Frederick Griffith in one of his famous experiments. What he did was he took two different species of Streptococcus, one being a virulent strain and the other non-virulent, avirulent. What he did was he injected one mouse with the virulent one, death ensued, used that as a control, and then he used the avirulent one. Mice were fine, but then he killed the virulent strain and had it in a serum as long as the non-virulent one injected it, and the mouse had died. Well, the implications of this was the genetic material present in the serum was able to be picked up and adapted by the non-virulent one. So, sort of like a quick evolution, I guess you could say, or a mutation. Another form would be transduction, which is facilitated by bacteriophages. Bacteriophages are viruses that primarily attack bacteria. What they'll do is basically a virus will attach to a bacterium, make its way into the virus or bacterium, replicate, and then lyse the cell or cause it to explode. And when it's replicating in there before it explodes, basically it's just DNA and a capsid, and the capsid will re-surround the DNA that's present in there, but sometimes it'll actually take up the broken down bacterial DNA, and when it goes for another cell, it'll incorporate that into the new cell, and it'll instead cause mutation in that manner. And then, more so what they're going, conjugation is another type. Conjugation is basically the closest thing that you get to bacterial copulation. Basically a tube will form between the two individuals and genetic material will transfer across and be incorporated that way. Most often this is by plasmids. Plasmids are additional genetic material that is present in the bacteria besides their normal genome. This will usually impart them with additional virulence factors, resistance, and other matters. So that's another way they can do it. Next, I will show how the scientists use this information for different applications. And that brings me to genetic modification. Basically, an adulterated overview of genetic modification is the scientist they'll find a desired gene. A gene being just like a sequence of nucleotides that engenders a specific trait. And they'll try to isolate that, and they'll put that in the plasmid because the plasmids are easier to work with, and then they can transfer that to the host. Or, more interestingly, like when they work with vaccinations, they can manipulate, say, a virus or just a bacterium to make it avirulent, but it'll keep its factors that signal the body to mount an immune response, because vaccines work in a manner where you have 3 lines of defense in your body. You have your skin, or your outer, basically a barrier, then you have a non-specific chemical response, then adaptive immunity, which is what the vaccines will work in. Basically, you'll have an attenuated virus which has been modified so it'll retain the same factors that signal your body to mount a response if it were to see that again, only since it's not pathogenic, you'll just be able to prepare in advance. They'll also do this with yeast more commonly, which is kind of interesting. So the yeast it won't actually harm the individual, but it'll prepare them for that. And despite all this research, there's still multiple misconceptions. The one most prevalent that I see the most is like Roundup Ready corn. Roundup Ready corn is like supposed to be resistant to Roundup, which is a chemical that acts specifically, or it's like selective toxicity. It acts on a specific metabolic process in nematodes, and since humans don't actually utilize that metabolic process, you could technically drink a gallon of Roundup if you wanted to, and no harm would ensue. Well, maybe like overconsumption of fluid, because you can commit suicide like if you drink too much water. So still admonish against that. But in review, I went over plasmids, horizontal gene transfer, some application, and then selective toxicity. And despite, I would just like to remind you that the plural of antidote is not always data.